Thanks for coming, everyone. So, welcome to this CAIDA talk today. We have visiting us Kameron Decker Harris from Western Washington University, just down in Bellingham. So he is in the computer science department, but does computational neuroscience kind of intersections with machine learning, graph theory type stuff, other applied math PhD things you would expect. Kind of nice interdisciplinary stuff. So he's here because he met someone, a computational neuroscientist who's at a place that I used to be. Actually, like half of our UBC ML faculty are alumni of a theoretical machine learning and computational neuroscience department in the UK, which the others aren't here right now, but Frank and Mijong also were there, in addition to me. So UBC ML should have stronger connections to computational neuroscience people than it actually does in practice. So hopefully we can kind of start working to make that a little more true, because there's lots of neat connections, as we're going to hear about today.
Cool. Thanks, Danica. Yeah, so I actually could give a whole other talk. Oh, I guess I made my video. Okay, now it should work. I could give a whole other talk about that history, but I'm going to gloss over it. But going back to the earliest days of neural networks, it was inspired by neuroscience, and I think throughout time that overlap has been pretty strong. So today I'm going to talk about geometry of neural networks. So basically function-space geometry, but with some structures that you see in biology and also in networks that get trained. So we'll dive right in. And Zoom land, if you guys need to chime in at any point, if it's not getting through, unmute and get our attention. Okay, so here's kind of the overview. So just a little bit of an intro about structure and randomness, and then we'll dive into some actual research that I've done on this stuff. So in real brains now, not artificial brains, there really isn't sometimes a clear distinction between what's random and what's not. In the brains of very simple brains of insects, for example, you do actually see from one animal to another certain circuits where exactly the same neuron connects to exactly the same other neuron reproducibly, like from one individual to another. So things like in Drosophila, which is the fruit fly, you can label individual neurons and that happens. But then you have other circuits, including in the fruit fly, so this area called the mushroom body, which does associative learning, where it seems like the connections are random, and people argue about how random they are really, but you could say they're pretty random, and that's also like what you do when you have an artificial neural network and you initialize it, right? You start out with something random. So kind of the hypothesis that I'm exploring here is that you have lots of networks, large networks, where things are random, but not just like completely not completely random and ignorant, of some sort of structure in the input or structure in that network.
Sorry.
Already a question. Good.
Yeah, I don't know anything about neuroscience, but when you say connection, you mean like some kind of chemical paths? What do you mean?
Right. So in an artificial neural network, right, you're probably familiar with those. Yeah. You have weights.
Right.
A matrix of weights. In a real neural network, neurons are physically connected to each other, and there's lots of different ways that could happen. There could be synapses where there's neurotransmitters that are sending a signal, or you could have electrical signals directly affecting the other cell. So I mean that kind of thing, like an axon and a dendrite, which are the parts of physical neurons that those weights correspond to. And the weights you could think about as kind of like the conductance for those different synapses.
Yeah. So ultimately, it's kind of could be abstracted away, like the actual neural network. What's happening.
Right. So an artificial neural network is abstracting away a lot of that complexity and kind of ignoring dynamics, for example, which in reality are important. But we'll work on that very abstract level here. Think artificial. So, okay, so learning and development could modify the structure in those distributions. Yeah?
So in group flow example, the connections are deterministic right from start.
In
Group flow, the first one.
in things that are like pattern generators that generate their like walking strides, you have like one neuron that fires to another neuron, and that causes their legs to alternate back and forth, and that's always the same way. Okay. All right. So now diving into some math. Okay. And we can do theory in those random regimes, which is another good reason to study them. Okay, so a lot of you may have seen this already, but I'll just go over it in case you haven't. So let's think about an artificial neural network now, right? So we have some input layer that I'm going to have the inputs be x, vector, and then you have some feature layer, and we're going to keep it super simple, just a 2-layer neural network, right? And there's some readout stage that occurs, but we're actually going to not think about that for the moment and allow that to vary later. But assuming we have this set up, right, the way you would initialize this network is you would draw these W weights from this layer to the next, following some distribution, mu. And so these are the weights we were talking about. So neuron I in this layer will have a vector of weights W, right, that it projects on the input onto, and then it passes it through some nonlinearity. Okay, good. So in a limit of many of those weights, right, you can normalize the dot product between these feature vectors for 2 different inputs. So I have an input x and an input x prime, right? And I form the feature vector phi, and then I compute those dot products. And if I normalize that by the size of the layer in a large width limit, that's going to converge to its expectation, right? This is just like law of large numbers for these functions, for these random vectors. And that's what we're going to call a kernel, K, that computes a similarity between 2 inputs, x and x prime. Any questions at this stage? How many of you are familiar with this room? Okay, only 1 or 2. Good. So the idea is when my weights are random, right, I have this random set of features, but when the width is very large, the randomness kind of averages out, and you have a deterministic object, this K kernel function, which is just like the kernels that you know from kernel methods, etc.
Just trying to understand, is the sum and average over different random values of the weights, or is each feature being sort of
Oh, good. So what's being averaged over here? So here, the inputs x, oh, this should be prime, sorry. The inputs x and x prime are not random. We're averaging over the weights. So sampling of the weights. So each I has a different random vector W that are I.
So you have a bunch of features, but you haven't actually figured out what those features are yet. You're sort of letting them evolve into different things that
You could sample a network of size 100, right? And you'd have 100 random vectors. And if you did it with a network of size 1,000, you'd be more and more acting deterministically as you get larger. I mean, you could get really detailed into what kind of convergence you care about. I mean, I'm kind of sweeping that under the rug here. Point-wise. Okay, you good? Okay, so you think about this, right, as an overlap, right, in feature space. And this goes back to the 90s initially. Okay, so why am I interested in this? Well, it's going to be easier to understand networks in this kernel limit. So you can think about doing learning with a random network as kind of like doing kernel learning. And, for example, regression is going to be like doing linear regression in some kernel space. And we know how to understand linear regression, for example. And so the one question would be, if I have different kinds of networks now, changes in the weights, changes in the nonlinearities, etc., how does that affect the kernel? Okay, so that's sort of the setup. So the first project I'll talk about is related to thresholds in networks. So a very simple thing, right? Like what's the threshold of my neurons? The equation I showed you before doesn't actually have a threshold, but we'll put that in. And this is with folks from Columbia University, and we have a preprint submitted. So we were inspired in this setup by another neuroscience system, the cerebellum. It's totally okay if you have no neuroscience background. I'll try to make it simple. But it's this place here in the back of your brain, and it's actually contains more neurons than the cortex. So people make a big deal about the cortex, but there's a lot more neurons in the cerebellum. They're just smaller and probably not doing as complicated stuff. But one thing that the cerebellum is important for is prediction of movement trajectories. So when you drink alcohol and you get tipsy, one of the reasons why your movements are less coordinated is because your cerebellum function is decreased. And people who have certain kinds of tremors, like this picture here, they're trying to draw this smooth line, but they're tremoring as they do it. That's known to be a cerebellar defect. So we think that something that it's doing is almost like motion control. And if you think about doing movements, usually those are some sort of like smooth movement. So that's one model for what the cerebellum is doing. It probably does a lot of other stuff too. Okay, so what's the network architecture? So the inputs to this network are carried by these things called mossy fibers. And so that's just the actual physical thing that carries the inputs. So that you can think about as the vector x. And then there's this layer of cells called granule cells that receive those inputs, and then they have their outputs project up into these parallel fibers, which are then read out by this neuron called a Purkinje cell. So one Purkinje cell will read from about 200,000 granule cells. Those are those very abundant cells. And then about 7,000 mossy fibers will be presynaptic to that. So that's kind of like the size of the network for one Purkinje cell. So we'll abstract this into that same picture again, right, for a two-layer neural network. Simple story. And the plasticity in this network really does only occur in the Purkinje cell parallel fiber synapses. So we know that really only learning is occurring at this readout unit. Okay. So again, we're going to have the same kind of equation, but now I added a threshold in, and it's going to be shared across all the neurons, but you can break that assumption. So this was inspired by this idea. It's been around a long time that somehow having the activity of cells in the granule layer be sparse, called sparse coding, is going to be useful for various reasons. So we wanted to ask, okay, so if I have this kind of network, what level, like if I change the threshold, I'll make more cells go silent because they'll have to be above threshold for their nonlinearity to activate. So what role does that have on the coding ability or the ability of this network to learn?
So that threshold is kind of determines the sparsity of the.
Yeah, it'll determine the sparsity on average of these features because this, I guess I didn't show this, but this H function is like a threshold linear type of unit.
Like a ReLU.
It's a ReLU.
Yeah. So that kernel K was anti-K kernel, like it wasn't like.
So this is the Gaussian process kernel. It's not the tangent kernel, but you can do all this theory with the tangent kernel. Or it's also called the random feature kernel. They're not that different though. Okay. So this in earlier tasks that people did, they did these like random categorization tests. So what people would do is they would draw random points in some high-dimensional space, right? And then they would say, nearest neighbors, I want you to classify things with the same label as these points, okay? And in that setup, what we saw in previous work was that a very sparse coding level was optimal for error. And there was theory that kind of explained that. But what we saw when we started modeling smooth tasks, so now our task we're trying to model looks more like this motion prediction thing that the cerebellum actually does. Actually, it's a Gaussian process, okay? So we can control the smoothness of the target function in that Gaussian process. we saw that the coding level that was optimal shifted towards denser representations, so more units active, as the task got smoother. Okay? And now there's actual data that shows that the cerebellum is probably less sparse activity than people thought. So the model seems to predict that. And we can even do like a more realistic kind of task where we actually have like an arm that's following physical equations of motion, right? And you're trying to control it, and you get the same sort of thing, even though it's not a Gaussian process. So how does that happen? Well, you can see it through the kernel, okay? So changing the threshold is going to change the kernel. So maybe ignore this for a second and look here first. So here I'm showing you the kernel for two inputs, x1 and x2. And in this setup, what it depends on is just...
What's the coding level?
Oh, sorry. Coding level is fraction of units active. So there's a map between threshold and coding level.
Okay.
It's kind of like a sigmoid.
What was the error level? Like how to measure?
How is error measured?
Yeah.
So for the regression tasks, this one and the Gaussian process, it was measured in just squared error.
So the task is, can you just describe the task?
So here you have this arm following some equations of motion, and the input to the network is this sensory motor state. So you're assuming the network somehow knows the current angular velocities of the arm, and then there's some control numbers that are plugged in, and its goal is to predict where the arm will be like a couple time points in the future. So it's trying to predict a nonlinear ODE. And then the GP task, it was just, in this one, it's just, we're sampling data uniformly on the unit sphere, and the target function is a Gaussian process on the unit sphere.
There's a question on Zoom also.
Can you read it for me, please?
Devansha, you could just unmute yourself maybe? Or let's see if we can hear you.
Yeah, for sure. My question is, I can see all those graphs that when the coding level is high, I can see that the error rate goes high. So is it correct to say that when I engage more neurons, or I add more neurons in my neural network, it will increase the error rate as in due to overfitting or something like that, or is it like some totally different phenomenon that I'm missing?
Okay. It's not that there's more, it's different than adding more neurons. So adding more neurons tends to make the error go down, so a bigger network. But the coding level being higher, so here I'm actually showing it only between 0 and 0.5, but you can go all the way up to 100%. But if it's 100%, for example, your threshold is like negative infinity, and the neurons are all linear, right? So in that limit,
Right.
you would expect kind of higher error, right, for a nonlinear task. So the interplay between error and number of neurons is kind of interesting. You can get double descent in that kind of regime, but here we're just kind of taking the limit of many neurons.
Okay.
Yeah.
But the coding level is equivalent to firing neurons, right? In this scenario.
Firing neurons, fraction of neurons that fire on average for many different inputs. So which neuron fires when I give it input 1 or input 2, it's going to change.
Okay. Thank you so much.
No problem. Okay, so here, yeah, coding level, I'm glad you asked that question. So here, 60% of the neurons are active, right? And you get this curve for the kernel, and the kernel is only depending on the dot product between inputs, just due to some symmetries, the fact that we choose weights with Gaussian. And it changes as I vary this. There's some intuition you can get from how many things are activated for. So if I have a lower coding level, right, the number of neurons that will be activated for this input x1 is like a smaller section of this sphere than for a higher coding level. So that's why you can see it decay more rapidly at low coding levels. So it changes the eigenvalues of this kernel, but we can actually prove that their decay rates are the same. So asymptotically, they're going to behave the same. And we can prove some other properties about these things, but maybe we can skip that for now. And so how can you use this kind of kernel properties to do theory? Well, we work with the spherical harmonics to do that. So this is just assuming your inputs are on the sphere. A nice basis to work with are the spherical harmonics. And they're going to work for any kernel like this that's dependent only on dot products. And so we think about the function we're trying to learn, this target function, which maybe is a Gaussian process or something, expanded in that eigenbasis. Okay. And there are theories that are out there that we didn't write, but we're using, that can predict the generalization error, for example, of a kernel method applied to a certain type of task. So the error depends on the coefficients of the target function, C. If they're bigger, it's more error because it's a bigger function to fit, right? And the eigenvalues of the kernel, which I'm calling lambda. And in certain regimes, where the theory should work, it does work here. So P is the number of training patterns that we use. So as your training data goes up, you have less error, and its dependence on coding level is nicely predicted by basically the eigenvalues dependence on coding level.
So it seems like from all the graphs, like we need to hit some optimal level of sparsity to achieve the lowest error.
Yes. So the optimal level of sparsity kind of depends on exactly the task, right? And that's the message that we were trying to get across. And the eigenvalues, it's because the eigenvalues of the kernel are varying with task.
And that also
with
depends on
coding
the basis you use, right?
The basis you use, but the fact that it's spherical harmonics.
Right, if you use a different basis, the way the eigenvalues look would be different, right?
Yeah. So the eigenvalues depend on the distribution of the data, as well as the kernel. And that's one thing we're sort of sweeping under the rug here. So the distribution of the data we assume to be uniform on the sphere, but because that lets us do theory. But in reality, it's definitely not, and so the eigenfunctions will concentrate their energy onto where the data is denser. But I don't know much about how to do that kind of theory.
Also, C alpha and lambda alpha are for the empirical risk minimizer functions,
the C alpha is the true target function here. So the thing I'm trying to learn is this thing. It has some coefficients in this basis.
so you basically bound those things because you don't actually know right?
Yeah, so if you were trying to do this for a task where you didn't cook up the task itself, like you didn't draw a GP, you wouldn't know C alpha. Maybe you'd make some assumptions about the smoothness of the thing you're trying to fit, and that would tell you something about the C alphas, for example. So this theory works pretty well, and it's multiple groups have worked on it and gotten these kinds of equations. The level of rigor is fairly high at this point, but there's some extra work people are working on to make it completely rigorous. So this is kind of comes from the physics literature. So what's the interpretation, of these theories? Well, we've kind of transformed a network, and in this case, we just changed threshold, very simple, into a geometry in function space question. So a task, for a neuroscience, you'd call it a task. For machine learning people, you'd call it a target function. So something you're trying to fit is a vector in that space. And that could be representing a regression, something you're trying to do, or a classifier. Right? So the network is giving you an eigenbasis for all L2 functions. So it's saying if I have my target function in some canonical basis, the eigenbasis might be different, or rotation. And it's giving you eigenvalues that scale the different axes. Some eigenvalues are bigger than others, and so some axes are somehow more important than others for the network. Right? So this is just linear geometry in function space. And whether the network is well suited for learning a certain task or not depends on the projection of its vector onto that eigenbasis. So if my lambda 1 is this bigger axis, my function f here is more aligned with the leading eigen axis. It's going to be easier to learn than this function g. And if you want to do rigorous bounds, you can get that in terms of the Hilbert space norm of that function f being smaller than function g. So this is kind of the picture I have in my head about why this network is better. One network might be better than another for a certain task.
It's like the SVM margin thing, which is inverse.
Yeah. Is the margin inverse with eigenvalues or something?
It's inverse with the norm of the vector. That's not what
Yeah, I know like with kernel classifiers, like classical results have something about the norm will appear, the error will be proportional to the norm.
Or like,
Or squared.
if you remember, the Rademacher complexity of the
Yeah.
class kernel functions is controlled by the norm.
Yeah.
There's some trace of the kernel in there too, but you could just balance the traces, and then the norms is the important thing. Okay, any other questions at this stage? I'm going to switch gears. All right, so that's kind of like, I guess, in a lot of ways, sort of standard, application of kernel theory to some network problem. Now I'm going to talk about a different kind of, it's just linear regression, right? I'm going to talk about something that I think is a little bit more creative, which is what happens if you think about networks with weights that are actually interesting and have some structure in them, rather than they're just Gaussians on a circle, on a sphere. And so this is work I did with folks from UW and a collaborator from Geneva Labs who actually gathered some neuroscience data that you'll see in a moment. And it's in press here. So what's a receptive field? So I won't assume that you know what a receptive field is, since you all are machine learning folks. But you may have heard of it. So in neuroscience, a receptive field is what is the stimulus that causes a neuron to spike. Okay. So if I have a neuron in a visual area, these original experiments were done in frog and cat. You'll see that they tend to fire when a certain kind of like bar moves across the stimulus space. So you'll have things like these little oriented bars. They look like little wavelets that these neurons are fire in response to. An interesting system that you may not have thought much about is this crane fly. Right now at my house in Bellingham, there are a lot of these crane flies around. So these are pretty big flies. They look kind of like mosquitoes, like giant mosquitoes. They don't bite, they're harmless. But they have these like hanging kind of pendulums on there that used to be wings. They evolved from wings. They're called halteres. And what the fly uses them to do is when it's flapping and flying around, and it can sense torques, because as it flies, if it rotates, they'll deflect because of angular momentum, right? So they have little sensory cells at the base that sense the deflections. And people have actually done an experiment where they wiggle those halteres and seen what kind of wiggles drive the cells best to fire. And you see receptive fields that look like this. So now these are temporal receptive fields that are sensitive to certain frequency ranges, basically. Yeah, question?
Just generally curious, how do they measure the brain signal or function when they move these?
Yeah, good question, Mike. So they'll have the fly mounted, and there'll be a rig where they have an electrode in the neuron that they're going to measure the firing, and then they'll have, I think they use magnets on the halter to drive it with like white noise stimuli.
Oh, it's measuring the neurons in the halter, not in their brain.
At the base of the halter.
I see.
Yeah. So your tiny, tiny electrode.
Sorry.
We don't know, like, some, for example, which action. I guess we do know, like different parts of the brain have different actions. If you want to measure the activity at that part of the brain, you have to direct your electrons to that part. And that decision becomes hard.
So this is just the measurement of the sensory kind of like input. You think about it as an input, and then the brain's going to use that later on. You had a question?
Sorry. Yeah, if I want to think about that abstract image, having a spike on a neuron is like really having a spike on the function of that weight or something like that.
Okay, maybe I'll go to the board just for a second. I don't know if the people on Zoom will see, but so we think an artificial neuron, right? You think ReLU as like the input-output function. So what is going on this axis, right, is the W transpose X, or input X. What does this number mean for a neuroscientist? Usually you think of it as like firing rate, so spikes per second. So this is like what we call rate coding. So it's like the neuron is transferring information the number of spikes that it's firing.
Maybe just to also say, like, neurons don't transmit like continuous electrical signal that increases or something. They do this like big spike in signal.
Yeah, I would assume you knew that, but I guess maybe not. But what they do is they fire information like this.
Mm-hmm. Yeah.
These could be like, I don't know, 7 milliseconds apart. So many spikes per second is possible.
So basically what we care about is being 0 or 1. So then there is a spike.
So yeah, in some regimes, these neurons are firing very few spikes, and so what matters is like, did they spike or not? Mm-hmm. But when you're thinking about them in this rate regime, you're kind of thinking about one spike doesn't matter that much. It's like how many spikes.
I see.
But different systems use different coding.
So there's a certain dynamic going on.
So here, they're driving these neurons. They're firing lots of spikes, measuring many spikes. Like here, you only see 4, but you did an experiment, you get thousands. And then you average all the stimuli before those spikes, and then you get this curve like this.
I see. So different colors are different neurons?
Yes, each of those is a different neuron. Same with these. These are real data from other people. Okay, so now let's model it. So again, we're just going to use a simple 2-layer network. The inputs are going to be time series or images. There's your MNIST classical image, right? But the receptive fields are going to be the weights. And why does that make sense? Well, the weights are basically the optimal filter that neuron is responding to. So if the input X is exactly the same as the weight, it's perfectly aligned, the dot product is going to be as big as possible, right? So the receptive fields really are estimating like a weight that a neuron has. And the idea is all those kernel theories that I've talked about, and everybody's way of normally initializing a neural network is just to draw weights IID at random from spherical Gaussian or uniform, maybe. So they would look like this as the receptive fields, which isn't what real receptive fields look like. They look more like this. And so we can model that as a Gaussian process in the input space. So the W's now are going to come from a Gaussian process. Everybody get that idea? So the C, the covariance function, is going to be not the item. So, okay, we can create like a 2-parameter family of functions that gives us receptive fields that look like the Haltier one. So you have some sort of frequency dependence, and so this is very high frequency or very low frequency, and then they tend to fall off backwards in time because these neurons don't have infinite memory. And we can feed in a time series kind of task where, say you have the network is supposed to distinguish between is there a frequency in this time series or not. here it's like, I think 50 hertz. And you see that when those weights are like tuned to the stimulus, so they're selected to that frequency, they have very low error with a small network, right? That makes sense. If the frequencies they listen to filter out the frequency that they're looking for, then that's incompatible and the error is high. And then, maybe frequency detection isn't very interesting. It seems like it should be really easy. So we added like a frequency XOR task that's more interesting, and it still does really well. So maybe a more complex example would be vision. And if you do visual receptive fields from actual mouse or from our experimental collaborators, they kind of look like wavelets or Gabor filters, but they're a bit noisier when you actually measure them. So again, we have a different 2-parameter family that models those receptive fields, and we can, with one parameter, we can change the size, and the other parameter changes the spatial frequency. And we use these in a visual task. So now, again, this is a network where the input to hidden layer weights are fixed and random, and then we're learning the readout to do a classification in this case. Having these structured weights, V1 inspired structured weights, which is the visual area, gives you a big benefit for this simple machine learning task, right? And besides just seeing that it gets you lower error with a smaller network, you can also show that in like few-shot learning type of setup, it does better. And also, like if you're going to train these networks now end to end, like with gradient descent, you do much better. And the point here was not to get the lowest error on MNIST or KMNIST. A convolutional network does much better. Is to understand how the weight structures actually matter. Yeah.
Just to be clear, for the previous one, what you did was the weights were trained off the receptive fields, and then we did tests on actual number data.
How did we actually pick the parameters of the receptive fields? Yeah, we actually did. I think we used the frequency and size parameters that we had gotten from the V1, like the neural data, and used it on these.
This is time series, or?
No, this was from. So how these were estimated, these V1 receptive fields, was by presenting lots of visual stimuli to a mouse and then seeing what caused the neurons to fire.
But this is like a 2D image.
2D image.
Yeah, and then
not time series. So you can make spatial temporal reception fields too that have both components, but we didn't do that.
So are those actually like the experimentalists did like a search over, choose a neuron, search over the space of images, what makes it fire the most?
Yeah.
Okay.
Yeah, it's a pretty standard experiment actually. So the collaborator we have has a setup where they put in this multi-electrode array that can record like 10,000 neurons at once, and then they do these experiments where they're just like flashing images at this mouse.
Yeah.
And they can do it anesthetized or not, and it makes slight differences.
Right. So, but that's a pretty difficult search space. Like
It's a large space.
Saying that the exact image you showed with the fuzz on it, neural measuring is kind of noisy. It's a very hard. It's not clear that
if the noise is
particular
due
kind
to
of noise is actually what that neuron likes necessarily, right?
I mean, you have a good point. But the experimentalists have like standard techniques for like measuring SNR basically in these cells. We excluded a bunch of the noisiest cells. These actually come out of a regression against the stimulus. And whether or not you do any penalization there, we'll smooth them or not smooth them.
So, but like the difference between one of these and like the most similar Gabor filter or whatever, is it clear that this is actually better for that neuron than?
We didn't do that exact test. Yeah, I mean,
this made it
yeah, you could really get into that if you wanted to. I guess I was interested in like, how can we put this into that random feature theory, right?
Yeah.
And so, lots of people have done the kind of things you're talking about.
Oh, sorry, there's a hand raised.
Sure.
Hi, I have just a quick question on this slide, so I understand
This one.
the picture of the neural network. So, this is a single layer, multi-layer version, like with only one.
That's right.
Okay. So, and the idea is that because it's a multi-layer version with one layer, it is supposed to work like a
Yeah, and you can make it work. You can do this kernel thing for multiple layers. It's really not any different.
Right.
Okay, so I want to get a little bit further ahead, and then we can go to questions. But yeah, please interrupt again. So I want to just point out that I did try this in like a deep network, AlexNet. And an interesting behavior we see is that like when you do training on AlexNet, so there's a huge, on ImageNet, right? So the classic thing, there's a huge data set. Initially, having these structured features does actually have you at a lower loss. But as you train, the classical kind of initialization eventually matches it. And in the end, actually, like after the 90 epochs, which is standard, the classical weights are slightly better. And it seems to be consistent, which is interesting to me. Somehow focusing on these low frequency kind of filters maybe doesn't let the network explore as big of a space. I'm not really sure why it's that way. But it also, in this setup, you know, you've got convolutions. In that other setup, we didn't have convolutions, which is a huge bonus for the U1 weights there. And many fewer of, like, this fraction of the network that's using the structured weights in this case is tiny compared to the other setups.
So basically we had just a one layer network in the previous example.
In the previous example, one layer,
Yeah,
one hidden layer.
I see. So in the way that you, sorry again asking this question, but the way that you choose the weights are based on that V1, which sort of input makes this weight higher the most. But
Yeah, or basically I wanted the weights to look like this. So rather than the weight just being like speckly noisy, I wanted them to have some low frequency structure and be localized in space. Just like a convolutional filter is localized in space. So let's get into a little bit of the math, and then I'll wrap up. So, right, the weights are drawn from this Gaussian process. Okay, so it's just a Gaussian distribution with a covariance matrix when you discretize space. So that has some covariance, right, which is C. And that can be diagonalized by a unitary matrix. And I'm gonna write lambda squared as the eigenvalues because they're positive or at least not negative. Okay, so if I want to sample, right, from a correlated Gaussian, all I have to do is take a Gaussian that's spherical, scale it, and then rotate into that new basis. So in the network, if I do W transpose and input X, well, it's the same thing as taking that input X and projecting it onto U and then lambda and then G, right? So in a sense, I'm working in some new basis, on some new coordinate space where my X changed from rotated into this X tilde, which is just U times X. And then I filter, I scale things up and down by my lambdas. So if it's a big lambda, it's going to pay attention to that component or it'll filter out some components. And you know, if you think about what's the kernel here, well, it's just a new kernel that's in this other coordinate system. And so for frequency detection, the idea was there, if I work in the Fourier space, that task had no information in most of the components of the time series, or most of the Fourier components. And there was one component where it had information about what to do. And at the frequency, I was trying to learn, I'm basically trying to do that, I'm trying to classify what's small or big in that space, right? So the optimal, target, the optimal thing to do is for my classifier to draw a circle somewhere like there. So my target function that I was trying to learn is something like a quadratic in that one frequency component. So I have to somehow know that the network has to somehow either learn or know a priori that it just cares about frequency in a certain bandwidth, a certain frequency band, and compute like a nice quadratic or something like a quadratic there. So you can do the kernel theory in that case, and basically what happens is if you work with the structured weights or not, you have a different Hilbert space. And if your structured weights are basically filtering out all but d prime components of the input, basically only that many lambdas are non-zero, then basically your norm is going to be exponentially smaller than in the other case. Because it knows a priori what space to use. Does that make sense? I'm seeing nods, but... Okay. So what's kind of interesting, I think, is that we know that kernels are bad at learning target functions like this. So one kind of thing that a network can do that a kernel can't do is learn a function that depends on a lower dimensional subspace, like projection pursuit type of thing. But a network is good at that. And these structured kernels aren't learning that subspace, but it's saying, okay, if I have that kind of weight structure in my network, it's going to do well on that kind of task anyway. So it kind of gives us a way to understand what kind of weights are going to be compatible with a task or not. I don't think these ideas are that new, but it's kind of like a new way of presenting everything with the kernel. And then I guess I'll just finish, that these are kind of an interesting way of fitting biological data to real networks. And there was this interesting paper where they kind of proposed exactly the same thing in parallel to us that I just realized a couple days ago when I found this. And we're doing some new stuff with those structured weights now in multi-layer networks, and it's similar to like these scale space ideas or wavelet scattering transform ideas that are out there. And we're trying some experiments with compressed sensing as well, and it seems to work. So yeah, I might as well just leave it here, and thank you for your time.
Okay, we have some time for questions, either here or on Zoom.
Yeah.
How do you impose the V1 inspired weight structure? Can you do that? The learning process is different from...
How do I force it to look like this?
Yeah.
So it's just a choice of that covariance function, covariance matrix. So if you want to see the details of that, it's just... Actually, I think I hit the slide. Maybe it's better. People on Zoom can actually see. So the idea is we have some data, and since it's 2D, if you vectorize it, you get this like banded structure. But you can measure the covariance of the real data, and then we have a model, which is a 2-parameter function, and it's just that like the covariance of 2 points in time, or 2 points in space, sorry, is like a complex, it's a squared exponential, and it's just like, you have a squared term like this, which makes it smooth at a certain wavelength, and then you have another term which makes it die off in each of the directions, and they have a different scale. So one term is forcing nearby points. So t is a point in space to have the same to be correlated, and then the other one is just so that it dies off as you go away from the center of the space origin. And with the V1 covariance that we have because it's squared exponential, we can actually like find its eigenfunctions, and they're Hermite wavelets. So these little wavelet type things are the analytical bases that those weights are built out. So you can think about using those certain weights as doing a wavelet transform first, and then filtering out really high frequency information.
Do you happen to have any insights between how, like, your model of cerebellum relates to Minerva's sparse distributed memory by chance?
Sparse distributed
Memory.
memory. In that sense, sparsity is referring to sparse activity, right?
I think
Or sparse coding.
the sparsity there was that you have few neurons that cover the space of all possible beta that can be stored in there. I guess it also translates into sparse activations. Yeah.
I don't know, honestly. I think it's a similar idea, but I'm not sure.
Okay. Yeah, I saw like this supposed like biological implementation of like sparse distributed memory, and
Is this the fly stuff that they were doing?
I don't remember if it was the fly stuff, but when you had the whole diagram, like the Purkinje cell and whatever, like the granule cell, like that was the exact diagram that was shown as a possible implementation of SDM. So.
Yeah. So a lot of people have made theories of these cerebellar networks, and that learning and memory center in the insect brain, the mushroom body, is kind of the same setup too. And one thing I didn't make a point about also is that there's actually sparse connectivity here as well between inputs and second layer, which I've studied separately, but I didn't get into that. But yeah, some folks have modeled that as actually doing like a locality sensitive hashing as well, which I think is maybe related to sparse memory that you're talking about. It's all very close, but it's like people use different languages to talk about.
So I had two questions. One was like, when you do this abstraction away from the actual thing that's happening, does that really affect too much in practice as
like
just ignoring
what are we
those
losing
dynamics
from
that are
the real
actually
brain?
happening?
The main thing we're losing in these systems is the dynamical part. So neurons are, responding temporally. I'm just looking at like a static input-output function, right? But real networks, like the inputs are changing with time, and the neurons are integrating that information over time and in nonlinear ways too. So like there's chaos effects. There's lots of stuff that's lost there. And these kernel theories, I've seen there's a recurrent study of the neural tangent kernel that relies a lot on discrete time, and I don't really understand it yet. So that's sort of a big thing that I think is missing. It's like, and if you're trying to do RL or something like with a dynamical system, you really need that temporal information.
Yeah. So another question was like, I was trying to put it all together, the presentation in my mind. Like, so you had this hypothesis that for this sort of design of network, you have to have a small fraction of the network firing, like.
Oh,
it
in
should
the
be-
first part?
Yeah.
Mhm. But we were basically showing that that was false here. That the network would do better with 30% of the neurons firing.
Threshold.
Yeah.
Yeah.
And the cool thing is that actually matches the more new recordings in cerebellum, where they see definitely more than 10% of the neurons are firing in relevant time scale. And probably what I'm actually would be interested in too is like different parts of the cerebellum do different things. Are the coding levels of those different areas somehow matched to task? I think that would be a way, that would kind of indicate that this is not, that the theory sense actually saying something meaningful there.
I see. So you mean we use a certain threshold in our activation function, we could change that depending on
Right, in real neurons, like they're modifying their thresholds
so there's
due to
a,
feedback or just homeostasis type of things.
so there's a cluster of networks having a certain threshold.
It's a question from David on Zoom.
Okay, go ahead, David.
Hi, thank you, Tom. We're equating real neurons with the prior, no data for initialization, which is the real neurons being trained. It's an evolution of trained and not just random initialization. There are trained neurons, which is wondering how trained neurons relate to the values.
Yeah.
Sorry, can you repeat?
So I think the question, I'm just going to repeat because it was kind of quiet here. The question is, in these real neurons do have experience either through some sort of feedback mechanism or evolution that causes their weights to not be, maybe not entirely random or like somehow trained. And so how does that fit with what we chose? So I think maybe it's most relevant for the second part of the talk, right? So here what I was doing was modeling these weights as following some distribution that has structure that looks realistic. So I guess one question would be like, is that even a realistic model? And I think I showed that it is fairly realistic. But then I think your question is maybe like, so how did that structure get in there? And it could be many different ways. It could be evolution, like you say, and somehow evolution got the animal to learn to develop connections in the visual areas that have this kind of structure. I think it's probably a lot that, and it's also unsupervised type of rules. So if you go back and read like the earliest neural network convolution papers, Fukushima's neocognitron, every layer was unsupervised. And in real networks like in brains, there's not backpropagation probably. So probably there's some sort of maybe unsupervised rules that are helping kind of sculpt these connections to have structures like I was talking about. Does that seem to answer your question?
I think so. I just said as though the analogy between the real neurons and the learned neurons of the prior.
Yeah, but in the system, in the cerebellar system or the mushroom body system, there really doesn't seem, there's no known mechanism for plasticity in that first layer that I modeled as random. And we were allowing plasticity in the readout layers. So
Plasticity goes in evolution, right? So evolution would have done, so it's useful.
True, yeah.
Sense, but random, relatively random sense.
Well, I guess the point I'm trying to make is that random is a spectrum, right? So if you actually look in the cerebellum it does have structure between different cerebellar regions, like where it gets inputs from and stuff like that. If you actually look in there and you try to do a test between is this a random network or not, I don't think you can show that it's not random in that situation. I think the best evidence for that is in the mushroom body, though, the fly learning center.
Thank you.
But yeah, I mean, you make a good point. It's definitely not entirely random. It's a mix.
Maybe, since we're a few minutes past 12 now, maybe we should call this the official end. But if you get more questions, just hang around for a couple minutes.
Thanks, everybody.
Thank you.
Nice to have a good discussion. Appreciate all the questions.